Thank you for the visit. SciQ is science made simple for the rest of us. Enjoy. Researchers at the Institute of Molecular Biology in Mainz, Germany, have made a breakthrough in understanding the origin of the aging process. They have identified that genes belonging to a process called autophagy, one of the cell's most critical survival processes, promote health and fitness in young worms, but drive the process of aging later in life. This research, published in the journal Genes in Development, gives some of the first clear evidence for how the aging process arises as a quirk of evolution. These findings may also have broader implications for the treatment of neurodegenerative disorders such as Alzheimer's, Parkinson's, and Huntington's disease, where autophagy is implicated. The researchers show that, by promoting longevity through shutting down autophagy in old worms, there is a strong improvement in neuronal and subsequent whole body health. Getting old, it's something that happens to everyone and nearly every species on this planet, but the question is, should this be the case? In a recent publication in the journal Genes in Development titled Neuronal Inhibition of the Autophagy Nucleation Complex Extends Lifespan in Post-Reproductive C. elegans, the laboratory of Dr. Holger Richley has found some of the first genetic evidence that may put this question to rest. As Charles Darwin explained, natural selection results in the fittest individuals for a given environment surviving to breed and pass on their genes to the next generation. The more fruitful a trait is at promoting reproductive success, the stronger the selection for that trait will be. In theory, this should give rise to individuals with traits which prevent aging, as their genes could be passed on nearly continuously. Thus, despite the obvious facts to the contrary, from the point of evolution, aging should never have happened. This evolutionary contradiction has been debated and theorized on since the 1800s. It was only in 1953, with his hypothesis of antagonistic pleiotropy, AP, that George C. Williams gave us a rational explanation for how aging can arise in a population through evolution. Williams proposed that natural selection enriches genes promoting reproductive success, but consequently ignores their negative effects on longevity. Importantly, this is only true when those negative effects occur after the onset of reproduction. Essentially, if a gene mutation results in more offspring but shortens life, that's fine. This is because there can be more descendants carrying on the parent's genes in a shorter time to compensate. Accordingly, over time, these pro-fitness, pro-aging mutations are actively selected for, and the aging process becomes hardwired into our DNA. While this theory has been proven mathematically, and its implications demonstrated in the real world, actual evidence for genes behaving in such a fashion has been lacking. This evidence has now arrived, according to the co-lead author of the paper, Jonathan Byrne. The evolutionary theory of aging just explains everything so nicely, but it lacked real evidence that it was happening in nature. Evolution becomes blind to the effects of mutations that promote aging, as long as those effects only kick in after reproduction has started. Really, aging is an evolutionary oversight. Jonathan continues, these AP genes haven't been found before because it's incredibly difficult to work with already old animals. We were the first to figure out how to do this on a large scale. He explains further, from a relatively small screen, we found a surprisingly large number of genes, 30, that seem to operate in an antagonistic fashion. Previous studies had found genes that encourage aging while still being essential for development, but these 30 genes represent some of the first found promoting aging specifically only in old worms. Considering we tested only 0.05% of all the genes in a worm, this suggests there could be many more of these genes out there to find, says Jonathan. The evidence for aging driven by evolution was not the only surprise the paper had in store, according to Thomas Wilhelm, the other co-lead author on the paper. What was most surprising was what processes those genes were involved in. Not content to provide just the missing evidence for a 60-year-old puzzle, Wilhelm and his colleagues went on to describe what a subset of these genes do in C. elegans and how they might be driving the aging process. This is where the results really get fascinating, says Dr. Holger Richley, the principal investigator of the study. We found a series of genes involved in regulating autophagy, which accelerate the aging process. These results are surprising indeed. The process of autophagy is a critical recycling process in the cell and is usually required to live a normal full lifetime. Autophagy is known to become slower with age, and the authors of this paper show that it appears to completely deteriorate in older worms. They demonstrate that shutting down key genes in the initiation of the process allows the worms to live longer compared with leaving it running crippled. This could force us to rethink our ideas about one of the most fundamental processes that exist in a cell, Holger explains. Autophagy is nearly always thought of as beneficial, even if it's barely working. We instead show that there are severe negative consequences when it breaks down, and then you are better off bypassing it altogether. It's classic AP, he continues. In young worms, autophagy is working properly and is essential to reach maturity, but after reproduction, it starts to malfunction, causing the worms to age. In a final revelation, Richley and his team were able to track the source of the pro-longevity signals to a specific tissue, namely the neurons. By inactivating autophagy in the neurons of old worms, they were not only able to prolong the worm's life, but they increased the total health of the worms dramatically. Imagine reaching the halfway point in your life and getting a drug that leaves you as fit and mobile as someone half your age, and then actually outliving those persons. That's what it's like for the worms, says Thomas Wilhelm. We turn autophagy off only in one tissue, and the whole animal gets a boost. The neurons are much healthier in the treated worms, and we think this is what keeps the muscles and the rest of the body in good shape. The net result is a 50% extension of life. While the authors do not yet know the exact mechanism causing the neurons to stay healthier for longer, this finding could have real-world implications. There are many neuronal diseases associated with dysfunctional autophagy, such as Alzheimer's, Parkinson's, and Huntington's disease. It is possible that these autophagy genes could represent a good way to help preserve neuronal integrity in these cases, elaborates Thomas Wilhelm. Assuming such findings could be recapitulated in humans, it does offer a tantalizing hope to prevent disease and get younger and healthier while doing it. Already, science and biotech are using gene therapy and gene editing to change the ways genes express themselves. Now science is identifying many of the problem genes that directly cause disease and old age. Identifying the problem genes and then having the tools to cause them to express themselves more usefully may create a clearer path towards healthy aging and even age reversal. If you enjoyed the video, please like, subscribe, share and leave your comments below. Thanks for watching. Stay tuned for more releases from PsyQ.